Alright, we're back here. This is Wolfman Jack with the music for the. Well, that's actually a really bad representation of what happened back there, Wolfman Jack days, which is before your time, so you would know the difference anyhow. So let's continue on with part 4 of the biotechnology's potential of improving human health. So, we have, of course, visual representation of the genome that was in the picture here. Understanding the phenomenon is nice, but controlling it takes the satisfaction to an entirely new level. This might explain why there's so much excitement surrounding the field of biotechnology, in which technology is used to modify organism cells and their molecules to achieve practical benefits. As we have seen in this chapter, the structure and function of DNA are pretty well understood. Biotechnology uses this knowledge to achieve practical benefits, such as developing medicines to fight viral infections, to produce more nutritious foods while reducing reliance on pesticides, and to analyze forensic evidence as part of our criminal investigations. So what is biotechnology? It is primarily genetic engineering, adding, deleting, or transplanting genes from one organism to another. The modern emphasis in biotechnology is on genetic engineering, the manipulation of an organism's genetic material by adding, deleting, or transplanting genes from one organism to another to alter the organism in useful ways. Such manipulation may create a better tasting tomato or improve a treatment of human disease. So the biotech advantages in human health fall into 3 categories: producing medicines to treat diseases, curing diseases, and preventing diseases from occurring in the first place. Wow, look at that face. Anyhow, we can see in agriculture, biotechnology is used to create more nutritious and healthful foods as a way to improve our lives. So we have 5 steps of biotechnology. We can chop up the DNA from a donor species, amplify it, or the small amount of DNA, to make more useful quantities, insert it into the different DNA pieces into bacterial cells. We grow them, separate colonies containing the different inserts, and then identify the colonies that receive those inserts. This is all part of various aspects of biotechnology, and we use bacteria as little factories. And so, as we can see, the list of things that we can do here. So, how do we create a plant resistant to becoming eaten by insects, or a colony of bacteria that can produce human insulin? All of these things can be done with biotechnology. So again, we chop up, amplify, insert it, grow it, and then identify the different inserts. If you were trying to identify a gene for insulin, which particular step of the procedure of the outline would you use? So if you were trying to identify a gene for insulin, you cut human DNA into small pieces with a restriction enzyme, amplify the DNA pieces with PCR, insert a gene into a plasmid and insert that into the bacteria, or you would probe a gene library. And if you pick number 4, that would be right. Probing would be trying to identify certain regions in the genome that are compatible with the insulin. So number 4 would be right. So using scissors, or molecular scissors, is another name for restriction enzymes. We can reduce decades of complex research down to 5 steps. We can chop, identify various genes, such as harvesting products. Interest may require taking the gene and inserting it into another species, possibly a crop plant or maybe a sick human. So the gene of interest is located on a section of DNA from the donor species. Restriction enzymes that target a particular base pair sequence on the other side of the gene are introduced. This example, the restriction enzyme recognizes ATCGAT and can be cut between the first A and T. And that enzyme binds the target base pair sequence and cuts the strand of DNA. The gene of interest has now been separated from the donor's DNA using these very specific sequences. And if you notice, we have ATCGAT, and ATCGAT is its complement, and the sequences are unique for that particular enzyme. So here's the summary of what I was just saying, that we can reduce decades of complex research down to these 5 steps. And so chopping, amplifying, inserting, growing, identify. And these are the techniques. So this may be as simple as harvesting a product of interest and taking the gene and inserting it into another species, either a crop plant or some sick human. To be sure, it's easier said than done that these steps can capture the essence of most of the modern biotechnology techniques. Not all of the steps are used in biotech, but only utilize one or a few of these techniques perhaps. But nevertheless, the mainstays of today's biotechnology world include some of these steps. So we chop DNA from a donor. To begin research, researchers select an organism that has a trait that they want. The researchers may want to produce human growth hormone in large amounts. First, it would be able to obtain the human DNA and cut the DNA into smaller pieces using restriction enzymes. Restriction enzymes have a single function. Whenever they encounter strands of DNA, they cut it up into small pieces. These enzymes evolved to protect bacteria from attacking viruses. By attacking from the viruses, so we can chew their DNA up. Immediately upon entering or encountering DNA from an invading virus, a restriction enzyme recognizes and binds a particular 4 to 8 base sequence and cuts the DNA, thus making it possible for the virus to reproduce within the bacteria. So we just chop up their DNA and shred it essentially. Dozens of different restriction enzymes exist. Each recognize a different sequence that cuts the bases. So those become our tools for manipulating DNA. So restriction enzymes are one of the key tools in biotechnology because it's the first step of nearly all biotechnology applications. Involves cutting the DNA from donor species that have a gene or genes of interest to shorter length. So the restriction enzymes cut DNA at particular places. It could be overhang or, in this case, a blunt cut, so that we have the same numbers as it cuts there. And so we also have other technologies, amplifying DNA pieces using polymerase chain reaction. We take a solution containing isolates from a segment of DNA, we heat it. The enzyme DNA polymerase is added along with a large number of nucleotides and then is cooled. The DNA polymerase adds a complementary basis to each single strand, and then the resulting we repeat over and over again, and we get amplified DNA as a result. Now, we talked about that in the lab. So amplifying DNA is into more useful quantities. In many situations, only a small amount of DNA will be available for analysis or other biotechnology use. PCR is a laboratory technique that allows tiny pieces of DNA, perhaps ones that have only been cut from a larger piece, to be duplicated repeatedly, producing virtually unlimited amounts of it. PCR is especially useful in forensics, since investigators often find only small samples of DNA at crime scenes, and then they can amplify it for DNA fingerprinting later. In the first step of PCR, the DNA of interest is heated to separates the double-stranded DNA into separate single strands. Next, the DNA is cooled. The enzyme called DNA polymerase and a large number of free nucleotides are added to the DNA mixture. Now, this DNA polymerase adds complementary bases to a single strand of DNA, causing the nucleotides to be added one by one to each of the 2 single strands of target DNA. This makes each of the single strand double-stranded again, resulting in 2 complete copies of DNA of interest, and they're identical copies. The process of heating and cooling can be repeated again and again until billions of identical copies of the target sequence are formed. And insert DNA by using plasmids. We can use plasmids to take genes of interest and amplify them using this technique. The target's or segment DNA is of DNA is isolated using restriction enzymes. Using the same restriction enzyme and a single cut is made into a bacterial plasmid, so we clone into that plasmid. So this is a cloning technique. We're cloning that gene of interest. Because the same restriction enzyme was used to isolate the segment of DNA and the cut plasmid, the 2 share complementary base pairs and fit perfectly together. The plasmid, now including the gene of interest, is inserted back into the bacteria cell. the genes on the plasma can be expressed in the same bacterial cell are replicated whenever the cell divides. And so the bacteria now are the machineries for us to amplify that segment of DNA, and then we can isolate that DNA rather easily. So inserting foreign DNA into the target organism. So most modern biotechnology applications involve the transfer of genes from one species to another, creating transgenic organisms that can carry DNA from the donor species. In human growth hormone, for example, the researchers may want to transfer the human growth hormone gene into a bacteria, E.coli, with the goal of having the bacteria to produce the human growth hormone. To create a transgenic organism, it's necessary to physically deliver the DNA of interest isolated from the donor species into the recipient organism. This delivery is often accomplished by using the plasmids, like we just mentioned, which are circular pieces of DNA that are separate from the chromosome that can be incorporated into the bacteria's genome. This is an effective strategy because following certain chemical treatment, plasmids can be structured that easily take up DNA and cut with restriction enzymes. So you can go back to look at that diagram there. Genes on the plasmid then can be expressed in the bacteria cell and replicated whenever the cell divides, and make as much of that as we want. So creating a gene library, growing bacteria colonies to carry all of this DNA. So we take large amounts of DNA, it's chopped up in restriction enzymes. It's sort of what they call like shotgun approach. We just cut and then have random insertions into the plasmids, and then we characterize this gene library of it that might be held by the bacteria. So growing bacteria colonies that carry DNA of interest, cloning. Once the DNA of interest has been transferred to a bacteria cell, every time the bacterium divides, it creates a clone, genetically identical to the cell that carried the DNA of interest. The term cloning describes the production of genetically identical cells, organisms or DNA molecules, a process that occurs each time bacteria divide. As a consequence, the numerous rounds of cell division, it's possible to produce a huge number of clones. This can be valuable if each of the clones is producing a valuable gene product. In a typical experiment, a large amount of DNA may be chopped up with a restriction enzyme. Each of the pieces is incorporated into a plasmid that is introduced into the target organism. A bacteria cell then, of all the bacteria cells, are allowed to divide repeatedly, each producing a clone of foreign DNA fragments. Together, all the different bacteria cells containing all the different fragments of original DNA is called a clone library or a gene library. So identifying a gene by using a DNA probe to locate genes of interest among millions of clones in a genetic or a gene library, the bacteria wash with a chemical that breaks down the DNA, making it single-stranded. A radioactive probe then is washed over the single-stranded DNA. The probe is short length of DNA that contains a sequence of base pairs complementary to the gene of interest. And that's the key. The probe has a way for us to detect if that DNA hybridized or matched, and we use radioactive probes for this. The DNA probe that binds to the complementary base pair sequence found in the gene of interest glows with radioactivity, thus allowing us to identify the gene easily. So we identify bacterial colonies that have received the gene of interest. Although a clone library contains a huge amount of potential useful genetic information, the information has no particular order. So we use probes and hybridization to identify which particular piece of DNA that we want out of the millions that have been separated in the clones in the library. So we want to find the bacteria that contain the genes that we have of interest. So if you recall that question, we want to probe and find that DNA first and identify that clone, and then use that to amplify. So finding the needle in the haystack is essentially what we're doing. The hybridization requires the use of DNA probe, which is a short sequence of single-stranded DNA that contains a small part of the sequence of the gene of interest that has had some of its molecules modified so that they can carry radioactive elements. Bacteria that carry the gene of interest are washed with a chemical that breaks down the DNA, making it single-stranded. Then the radioactive probe is washed over the single-stranded. Bacteria with DNA containing the sequence complementary to the probe will bind and will find it because it glows with radioactivity. Once the desired clone in the library is identified, then these cells can be grown in large numbers. For example, vats of E.coli that produce human growth hormone. So which the following changes, base substitutions, insertions, are underlined in bold, which would have the most severe effect? We've already answered this one, but I just want to see if we can retain this information from previous. So the answer was 3 because it was mostly truncated there. So every now and then I try to bring back memories from yesteryear. Biotechnology is the use of technology to modify organisms, cells, and their molecules to achieve practical benefits. Modern molecular methods make it possible to cut and copy DNA from one organism and deliver it to another. Biotechnology has achieved some important successes in medicine, agriculture, and forensics. Treating diseases and producing medicines with biotechnology, we can cure diseases and treating diseases. For example, the treatment of diabetes. We can't always get what we want, though, and biotechnology would prevent humans from ever getting debilitating diseases, but the next best thing would be to cure them. So biotechnology often is directed at the more practical goal of treating diseases, usually producing medicines that can help treat these by producing these effectively and cost effectively by using biotechnology methods or treatments. So here's taking insulin that was produced by biotechnology as an example. So diabetes is a chronic disease in which the body can't produce the chemical insulin, which breaks down sugar in the blood. Complications from diabetes can include vascular disease, kidney damage, and nerve damage. As recently as 1980, if you were one of the approximately 15 million Americans with diabetes, each day you would treat the disease by injecting yourself with insulin extracted from the pancreas of cattle or pigs that had been killed for meat. For most diabetics, insulin injections kept the disease under control, but traditional process of collecting insulin this way was difficult and costly. Moreover, the cow and pig insulins differ slightly from human. About by 5% of the diabetics had negative reactions to the cow and pig insulin preparation. So having the proper one grown using biotechnology was a good thing to do. So why do some bacteria produce human insulin? We can use recombinant DNA technology. So Bob Swanson joined scientist Herbert Bayer to transfer the potential of recombinant DNA technology, both of which got the Nobel Prize. In doing so, they started a biotechnology revolution working with Stanley Cohen. Swanson and Bayer used restriction enzymes to snip the human DNA sequence that codes for the production of insulin. Then they inserted the sequence into bacteria we call E.coli, which we've talked about before. After cloning the new transgenic bacteria, the team was able to grow vats and vats of the bacteria, which turned out or produced the human insulin. The drug could be produced efficiently in huge quantities, which made it available for the diabetics. This was the first genetically engineered drug approved by the FDA and continues to help millions of people every day. So perhaps even more significant than providing a better source of insulin, Swanson, Bayer, and Cohen's application of biotechnology revealed a generalized process for genetic engineering. It instantly opened the door for more effective methods to produce many of the different medicines and treating diseases. Today, more than 1,500 companies work in biotechnology and produce products, about $40 billion or so in revenues each year. So important achievements produced by the development of insulin-producing bacteria would be the human growth hormone and erythropoietin, the ability to make red blood cells. So human growth hormone produces the pituitary gland. Growth hormone has dramatic effects throughout the body. It stimulates protein synthesis, increases utilization of the body's fat for energy to fuel metabolism and stimulate growth in virtually every part of the body. Insufficient growth hormone production usually is due to pituitary malfunctioning leading to dwarfism. When treated with supplemental HGH, individuals with dwarfism experience additional growth. HGH is also used to combat weight loss in AIDS patients. Until 1994, it was prohibitively expensive because it could only be produced by extracting and purifying it from pituitary glands of human cadavers. Through the creation of transgenic bacteria, or cloned bacteria, using a similar technique used in the creation of insulin-producing bacteria, human growth hormone can be now created virtually unlimited supplies and given to the people who need it. And so the availability of growth hormone can increase strength and endurance and may be irresistible tempting to some athletes, even at $7,500 a month supply. Recent sporting scandals suggest the illegal use of HGH occurs frequently among elite swimmers, cyclists, and other athletes. Erythropoietin, produced primarily by the kidneys, is also known as EPO. It regulates the production of red blood cells. Numerous clinical conditions such as nutritional deficiencies and lung disease and others, such as chemotherapy, can lead to anemia, a lower than normal amount of red blood cells, which reduces an individual's ability to transport oxygen to tissues. Cloned in 1985, recombinant human erythropoietin, or RUH-EPO, is now produced in large amounts in hamster ovaries and is used to treat many forms of anemia. Worldwide sales of EPO exceed $10 billion in 2004. That's a lot of money. In other words, we can reduce decades of complex research down to 5 steps: chop, amplify, insert, grow, and identify. Then we're ready to utilize the products. This may be as simple as harvesting a product of interest, or may require taking the gene and inserting it into another species, probably a crop plant, or maybe a sick human. To be sure, it's easier said than done, but all these steps capture the essence of the modern biotechnology evolution that we're really experiencing. Not all the steps are used, but most of them are. Restriction enzymes are used to isolate the genes of interest is really the big part. And again, chopping DNA from the donor organism using restriction enzymes. We've talked about this. The restriction enzymes have a single function. When they encounter strands of DNA, they cut it up into pieces. Chopping the pieces of DNA with the unique restriction enzymes are only one of the key tools used in biotechnology, because the first step of nearly all biotechnology applications involve the cutting of DNA from donor species. So what is blood doping? How does it improve some athletes? EPO has been the center of blood doping scandals by professional cycling. Why might cyclists be interested in having this? Because EPO increases the oxygen capacity of blood. In other words, we have more red blood cells. And this gives them the advantage of having the increased red blood cells. The blood now can be much thicker, which can actually deliver more oxygen, so you can have higher development or have more muscle exertion, but it can cause that thickening of the blood and cause the risk of heart attacks. So there's a downside to everything. Biotechnology has led to some notable successes in treating diseases, as we've discussed. Curing diseases, gene therapy, and the correction of malfunctioning genes. Now, this was a controversial area for many years, and it led to a lot of people dying from various effects, and as we were understanding the technique, but it is now used for life-saving therapies. The case severe combined immunodeficiency was worth examining in some detail, because it proves that it's possible to use biotechnology to exchange working genes for malfunction ones, and that permanently causes or cures, a disease in an individual. So severe combined immunodeficiency is a genetic disorder. A baby's born with the immune system unable to properly produce a type of white blood cell, leaving the baby vulnerable to just about everything. The disease usually causes death within the first year. There was a big story around the news for a long time about a bubble boy, because in the 1970s the boy suffered from this severe combined immunodeficiency, or SCID, survived for 12 years by living in a sterile germ-free bubble. He didn't need to do that nowadays because we have ways to genetically use gene therapy. Stem cells are cells that have the ability to develop into any type of the cell in the body. Researchers have been able to cure the disease in some individuals using gene therapy that is simple in concept but difficult in practice. First, the researchers removed some cells from the baby's bone marrow. These were stem cells, cells that have the ability to develop into any type of cell in the body. In bone marrow, they normally produce white blood cells, and individuals with SCID have malfunctioning gene that disrupts the normal white blood cell production. In a test tube, bone marrow stem cells are infected with a transgenic virus that carries the functional gene. If all goes well, the virus inserts the good gene into the DNA of the stem cells, which are then injected back into the baby's bone marrow. From there, the cells can produce normal white blood cells, thereby permanently curing the disease. So gene therapy has had a poor record of success in curing diseases because we have to use viruses. And sometimes those viruses can reconstitute, because most of them that we used previously were cancer-forming viruses, ones that would transfer DNA into ours. So gene therapy's difficulties encountered in several different areas, usually related to organisms used to transfer the normal functioning gene, i.e. viruses, into cells of persons with genetic disease. Gene therapy difficulty getting the working gene into the specific cells is needed. This difficulty is, for an example, gene therapy for cystic fibrosis, a disease in which thick mucus accumulates in the lungs. Attempts to use viruses like those responsible for common cold to insert the normal gene into the malfunctioning lung cells. Unfortunately, because the lungs have evolved to keep foreign objects out, the body is very effective in mounting immune responses against viruses used to deliver the working gene into the living lung cells. Difficult in getting the gene into enough cells and at the rate that actually would have a physiological effect. To effectively cure a genetic disease, it's usually necessary to get the functioning gene in a large number of cells to get the new gene to make the right amount. So the treatment apparently caused leukemia due to the problems with the insertion of the gene disrupting normal functioning of the white blood cells, triggering them to become cancerous. So problems with transferring organisms to get them into the cell sometimes produced unintended consequences. So viruses can cause serious side effects if they infect cells other than the target cells. And this was a real problem for a long time. So other gene therapy difficulties is the difficulty in getting the working gene into enough cells to have a physiological effect. Problems with a transfer organism getting it into the unintended cells. Gene therapy has had a poor record of success. The stem cells primarily from technical difficulties in transferring normal functioning genes into the cells of persons with a genetic disease. So intervening to prevent diseases using biotechnology, focusing on answering some questions. Is a given set of parents likely to produce a baby with a genetic disease? So we started screening so we can find various high probabilities of the incidence, like in Tay-Sachs disease. So many genetic diseases occur only if individual inherits 2 copies of the disease-causing gene, one from each parent. This is true for Tay-Sachs, cystic fibrosis, and sickle-cell anemia. Individuals with a single copy of the disease never fully manifest the disease, but might pass it on to their children. Consequently, 2 healthy parents may produce a child with a disease. In these cases, it could be beneficial for the parents to be screened to determine whether they carry a disease-causing copy of the gene. Such screening, combined with the genetic counseling and testing of embryos following fertilization, can reduce the incidence of the disease dramatically. This has been the case with Tay-Sachs disease, for example. Since screening began in 1969, the incidence of Tay-Sachs has been reduced by more than 75%. Will a baby be born with a genetic disease, a cystic fibrosis, sickle-cell anemia, Down syndrome, and others? Well, once fertilization occurs, it's possible to test the embryo or developing fetus for numerous genetic problems. Prenatal genetic screening can detect these types of diseases such as the cystic fibrosis, sickle cell, Downs, and others. The list of additional conditions can be detected is growing very quickly. To screen a fetus, it's necessary to sample some of the cells with from the amniotic fluid. This is what bathes the baby. And a lot of times, these carry chemicals produced by the developing embryo as a result of the metabolism of cells. So we use a technique called amniocentesis or chorionic villus sampling that we'll explore later on. But once collected, the cells can be analyzed by using a variety of means for that. In an individual likely to develop a genetic disease after life, later in life, not afterlife, later in life, if we could do that after life, it would be kind of a waste of money, I guess. Breast cancer, prostate cancer, and skin cancer, and all of those we run at a higher risk at all times. So DNA technology can be used to detect disease-causing genes in individuals are currently healthy but are at increased risk of developing later. So we use early detection for many of the diseases like breast cancer, prostate cancer, and skin cancer to increase our ability to treat that disease. Ethical dilemmas, discrimination, health insurance, how to proceed with this information. Well, biotechnology may reduce suffering and incidence of disease, but these potential benefits come with potential costs, including these ethical dilemmas. People who have had a gene that puts them at an increased risk of developing a particular disease, for example, might be discriminated against if they're not currently sick and may never suffer from the disease for which they have the heightened risk. Currently, only half of the states in the United States prohibit genetic discrimination. Insurance companies have already canceled or denied health and life insurance coverage upon discovering that an individual carries a gene that puts them at a risk of a particular disease. Additional parents who discover their developing fetus develop a painful, debilitating, or fatal disease soon after birth are confronted with a difficult question of how to proceed. So biotechnology allows us to identify whether a given set of parents is likely to produce a baby with a genetic disease, whether a baby is likely to be born with a genetic disease, and whether an individual carries a certain disease-causing gene that may have other effects later in life. So these tools can help us to reduce suffering and the incidence of diseases, but these also come with a potential significant cost or particular risk of discrimination. So I guess we've finished this section. We're going to move on to part 5 in the next part of the presentation. So study this section. This is an important one. This is one that you should be aware of. I'll talk to you again in the next section.